So in the last class, we have mentioned about different reactor systems, and these configurations are based on the cell density in the reactor. So either you can have cell suspension, and or you can have some biofilm reactor where essentially in a smaller volume you can get higher biomass concentration. So biological systems are inherently slower than chemical reactors. So whenever you try to do any chemical reaction, maybe you can achieve completion of the reaction in few minutes, whereas biological system typically would take hours to days. So aerobic systems or the bacteria or microorganism which are following aerobic metabolism, they're little bit faster growing bacteria compared to anaerobic organism. So when you need to use anaerobic systems, growth rate is slow. And in the first class, I have mentioned that in biological treatment process, our intention is to get higher reaction rate. So if you have issue with mu, then that should be compensated by X. So if your mu is very low, then you need to have higher X. And for that reason, in anaerobic reactor system, we prefer to use this attached cell system because you get higher biomass concentration. So essentially, even if you have very low mu, still X concentration is quite high. That's why you get reasonably good reaction rate or conversion of your substrate. Whereas in aerobic system, their mu is reasonably fast compared to anaerobic system. And there also you keep moderate concentration of X. So typically in suspended cell system, when you use suspended like anaerobic you have anaerobic contact process, and suspended cell system for aerobic you have activated sludge process. So for this kind of reactor, when you try to inoculate, even in any other bioprocesses also, forget about wastewater treatment, even in other bioprocesses where you use CSTR type reactor, here for aerobic, the initial X0 or inoculum concentration could be in the range of 10 to 20% of total reaction volume. Whereas for anaerobic, the initial biomass concentration will be 20 to 50% of total reaction volume. So you can see the difference. For aerobic, you need a little bit less concentration of initial inocular, whereas in anaerobic, you need higher concentration because total biomass concentration is the requirement of biomass concentration is more in suspended cell system. Whereas attached cell system, you have flexibility because you get much higher concentration in a given volume. And that's why for aerobic treatment system, all these suspended cell systems are preferred over attached cell system. Whereas in aerobic system, suspended cell systems are still preferred because it is easy to manage. And the design wise, activated sludge process, it is actually CSTR with cell recycle. It's one of the simplest design, and startup period is also very less because you can easily just set up the reactor and start operating the reactor without having much delay. So when you talk about configuration, in suspended system, different kind of configuration you can see. We have mentioned about two different types. One is activated sludge, another is sequencing batch reactor. But apart from that, also you can get many other configuration. And for attached cell system, trickling filter and RBC, rotating biological contactor, they're widely used. And also you can have some specific cases, you can have fluidized bed reactor. So in fluidized bed reactor, you need to fluidize the granule. Only the problem is in aerobic system, the granules are not very compact. They're a little bit fragile and sensitive to shear stress. That's why in fluidized bed reactor, aeration is done in a separate chamber. So suppose this is your fluidized bed reactor. Here you just fluidize the granule by maintaining certain outflow velocity. But before sending the liquid to the reactor, you just aerate the liquid in a different chamber. So the collision between air bubble and this fluidized biomass can be avoided in the reactor. So you just aerate the liquid and then send the liquid to the reactor to get treatment, aerobic treatment. So essentially, when the liquid will come in contact with the biomass, biomass will utilize the dissolved oxygen, and they will also convert the substrate to a different kind of product. We'll discuss about this anaerobic contact and high rate anaerobic process. And in anaerobic reactor also, upflow anaerobic sludge blanket, that is one of the widely used design. And hybrid anaerobic reactor, that is a modified version of this UASB. So we'll also show you what kind of design we use for these configurations. So activated sludge, as I mentioned our last class also, that you have a CSTR and a settler, and from settler you just recycle certain amount of sludge to the system. And as you are returning that sludge back to the reactor, that's why it's called return activated sludge, because the sludge is already active, because you're separating the active biomass in the settler and then returning the active biomass. So that's why it's called return activated sludge. Certain amount of sludge you need to bleed, because the entire amount cannot be sent back to the reactor, because you need to maintain certain F by M ratio. And after the sedimentation tank, you get from the top of the sedimentation tank, you get the clear liquid. So one of the major drawback of this particular process is the sludge generation, because you need to use appropriate measure to treat this waste sludge. And also aeration is very expensive. So when you use this kind of system, almost 80% of O&M cost, means operation and maintenance cost, will be contributed by this aeration. So aeration is very expensive for this particular system. When you talk about design, it is very easy to build, because you have a simple design, a tank followed by a settler. And at the bottom, you can just have various kind of sparger options. You can have membrane sparger, you can have ceramic membrane, you can have nozzle sparger. So activated sludge process is a very have the simplicity in design, very robust, very easy to operate, and downtime is very low, but maintenance, that operation cost is quite significant. Second option is the contact process where you have, so this is a attached cell system or biofilm reactor where you have some disk, and this disk will be submerged in the liquid. So whenever it will rotate,
Sir, you are not audible.
can you hear me?
Yes, sir. Now we can.
Okay, so this is the tank, and here you have the disk. So you can have complete submergence, you can have partial submergence. Now, whenever you rotate it, and this tank is open, so whenever you rotate the disk, biofilms, you can see there in the disk that yellow color biofilm form on the disk. So whenever you rotate the disk, for a certain period of time, it will get submerged in the liquid. So it will get exposed to the liquid. And then again, it will come out of the liquid area and it will get exposed to the air. So periodically, the biomass will get exposed to air and liquid. So whatever substrate will come in contact with the biomass, that will get utilized while it will rotate at very, very low speed. Generally, the rotational speed will be less than 10 RPM. And this is a lab scale reactor system, but in industry, larger scale, you will actually get very big drum kind of configuration. So it will have a very big drum, but only thing is this drum is actually not like a continuous drum. You have stack of dishes. So in between this multiple dishes, you will actually get some space for accommodating the biofilm. And this entire drum-like structure will rotate in a big tank. And submergence is also depending on the bacteria or biomass you are growing to maintain the submergence. Means if you require more amount of oxygen and less contact time with the liquid, then you have less submergence. If you want more exposure to the liquid or contact time with liquid, you just have higher submergence. And whenever it will rotate, it will also force the oxygen to get dissolved in the liquid, because the oxygen displacement method will actually help to increase oxygen concentration in liquid due to the rotating motion of this disk also. So this is
Sir,
one.
but sir, this disc have some kinds of blades in welded in them, or they are just a simple circular disc. Sir, in that case, like if it is just simple circular disc, then how will it be able to merge us like oxygen and etc.
Uh, they're mostly, whether you call them a blade or a disc, configuration will not be very different. Blade doesn't mean that it has a metallic component. So you can have mostly this when you use this kind of material, they're either made up of some polycarbonate or polyacrylic material. Sometimes you can use some low density polyethylene, because when you try to rotate the entire, the cylinder is composed of stack of dishes. So ultimately, if you see the cylinder, their multiple dishes are kept parallelly. And if you use any metallic disc, obviously the weight of entire cylinder will be quite heavy. Whereas if you use any lightweight material, which are also good for growing biofilm, then it will be easy for you to rotate it. And when you rotate it, as it is, as I told you, even in this picture, you can see the disc is actually not completely submerged in the liquid. So whenever it will come in contact with the air, then the biofilm will also get exposed to air. And mechanically, when you rotate the disc in any given liquid system, it will also create churning motion in the liquid. So whatever air is present just above the liquid surface, it will automatically get more interaction with the water, and that interaction will actually help to dissolve more oxygen in the aqueous phase.
So, sir, biofilms are placed on this these plates.
Biofilms are grown on the disk surface.
Okay, sir.
Like here you can see the yellow color biofilm. There actually the disk is actually transparent. So if you remove this biofilm, you will get actually a transparent disk here. So in the laboratory scale, you can use transparent acrylic material like plexiglass. So here the discs were made of acrylic material, and then you got this yellow color biofilm form on the disc. And to get
The rotation speed should be very slow, right? Because if we rotate it very fastly, then the culture may be disrupted.
yeah, if you rotate it very fast, right, then sloughing of biomass will happen. Means the biofilm will dislodge from the surface. That's why rotational speed is maintained at very low speed. So in large scale reactor system, around 5 rpm they maintain. So the rotational speed will be less than 10 rpm for this kind of reactor.
Sir, it's better that if you can provide some like what is biofilm, like we have not read yet about biofilms more.
Okay. Okay. So, okay. So if you can remember in the first class, we have mentioned about preservation of culture or growing the culture in the test tube. So you have some liquid media, solidified media, means all nutrients plus agar base is used to make this solidified media. Now you put your bacteria to grow the bacteria on the surface. And when you grow them, essentially bacteria will utilize the nutrient and form small colonies. So after getting this colony, you just take few colonies from there and then put it in a conical flask where you already have your media solution. Media solutions means essentially this solution will have all nutrients for growing the bacteria. Now you put this inoculum or the few colonies of culture using a sterile loop, and then you maintain optimum condition for this growth of this organism. So at a particular temperature and rotational speed, you just maintain the culture, and after a certain period of time, turbidity will also increase because you will get more biomass grown in the culture. Now, in this system, all this biomass, they are actually representing small solid particles. So essentially, if you talk about a single bacteria, you will not be able to see single bacteria using a naked eye because they're tiny creatures. Only the dimension of this bacteria will be in micron range. Now, when you have multiple bacteria uh, in a smaller place or smaller space, then they will form bigger granule. And if you get some flocks or granule having size of millimeter, say 1 millimeter or 2 millimeter diameter, then you can easily see them. So whenever you have high concentration in a smaller volume, multiple bacteria, they will try to collide each other and they may form these flocks. Now this sorry, whenever they will form the flocks, then essentially they will just form little bit bigger granules. Then you can see them. So one option is to use this method to get suspended cell, where you can have some idea that okay, bacteria are actually suspended in the liquid, and they're very tiny in size. That's why you couldn't see them in naked eye, but the concentration is there because you can see the turbidity. Because initially the media color will be almost transparent, and due course of time you will actually get uh, certain amount of turbidity, and that will signify that you got a lot of growth of bacteria. So these are all suspended bacteria in the liquid. So when we talk about suspended cell, that means we are talking about individual cell are, or maybe clump of cells are actually suspended in the liquid. And when you use CSTR, like the reactor where you have stator, in CSTR, you essentially impart certain amount of energy to the liquid to make this solid particle suspended in the liquid. So whenever you put that extra energy, solid will inherently have a tendency to get settled at the bottom due to gravity. Now, when you use this external force to suspend them in the liquid, definitely you will not have any uh, settlement at the bottom. You will just get all the solid particles suspended in the liquid. So in CSTR type of reactor, we deal with suspended cell environment. Now another environment where deliberately we don't want to create this cell suspension condition. So we don't provide any extra shear in the liquid. We don't use any vigorous mixing in the liquid. We just keep, suppose in similar fashion, conical flask, I just put few plates, and these plates are uh, interlinked. Means you have option to actually keep them in a static position. Now you keep the same media and also similar or different kind of bacteria, and here you just maintain temperature, but you don't move it. Means you just keep it in a static bed. You don't use a rotational movement of the shaker. Now whenever you have growth of bacteria, they will try to create film on the surface of this support material. So when you try to see the film, it will look like uh, a slimy layer is covering the slimy layer is trying to cover the surface of this solid plates. So essentially here you are not maintaining any shaking. That means you are not inducing any external shear force, and the bacteria which actually they are present in the liquid, they already got a surface to grow, because few bacteria they have a tendency to form this kind of uh, film. So essentially one bacteria will get attached to the surface, another bacteria will come that will also get attached to the surface. So first it will form a monolayer. So on the surface of the solid part material, you will have a monolayer formed by the bacteria. On top of the monolayer, you will have another layer of bacterial film. So in that way, you can have multi-layer bacterial film. So film means again agglomeration of bacteria, but they're arranged in a way that it will just form a specific layer of film, and that will have a specific thickness. Now this thickness will also depends on the local environment. Like here, you can see this extreme, the last disc has a little bit more thicker biofilm compared to the subsequent disc. So it is very much possible that the biomass uh, got attached to this surface is experiencing less shear. That's why they are not getting detached from the plate, whereas other plates are actually getting experiencing little bit more shear. That's why after certain thickness, it will automatically get detached, because whenever it will rotate, it will encounter minimal amount of shear. And if the thickness is significantly high, then the shear rate can impact the multi-layer biofilm at different levels. Suppose you have really very thick biofilm. They are not very compactly attached to each other. All these layers are not very tightly attached to each other. So suppose you are getting a shear stress, which will create little bit of detachment between third and second layer. So it is very much possible that when this entire biofilm will keep on experiencing similar amount of shear stress, after certain period of time, it will first generate a crack, and then ultimately this upper layer will go off. So in biofilm type of reactor, shear stress is a very important factor, and also the kind of support material you are using. Sometimes deliberately use some support material which will provide some electrostatic attraction. Suppose the biofilm are negatively charged, so you just use a positively charged inert material, a support material, which will essentially create or establish an electrostatic attraction. Sometimes
Sir, how will be the biofilm negatively charged?
most of the bacteria, they are negatively charged. Surface charge, if you check, most of the bacteria, they have a negative potential in the surface. So
Okay.
now, apart from the charge difference, you can have corrugated surface. Instead of smooth surface, you can have corrugated surface of the support material. So obviously, if the surface is rough, you will have more um, friction between biofilm, and so detachment will be a little bit difficult. With smooth surface, you have higher chance of detachment, whereas in rough surface you get lower chance of detachment. So different kind of mechanisms are used to get stable biofilm. You can use different kind of material also, which will essentially help longer attachment of biofilm to the inert support. And sometimes we also have reactor where we don't use any inert support. We get biofilm. Suppose you have a system where one few bacteria, they uh, they come together and then form a small granular structure, and on top of that granule, another layer of biofilm will be produced by some other bacteria. In that way, you can have multi-layer. So at the inner core, you have some bacteria, and outer layer also you have different bacteria. So essentially, in this granule, the entire granule is actually composed of by bacteria only. So these are called self-immobilized granules, where the bacteria will immobilize by forming granules, and typically they're very compact in nature. Another option is to use some inert support material, and on top of the support material, you will have formation of biofilm. So you can have sand particle, you can have activated charcoal particle, you can have different polymeric granules. So difference between these two is, here you have the entire granule is composed of bacteria, whereas here you have inert at the core, inert support at the core, and then surrounding that support material, you have biofilm. Both the system has certain advantages, disadvantages. We'll discuss that when we'll discuss about this fluidized bed anaerobic reactor. So rotating biological contactor is actually exposing the biofilm to the liquid due to periodic motion. Sorry, due to continuous motion, it will periodically get exposed to liquid and air. And when you use anaerobic rotating biological contactor, instead of keeping this top portion open, you just put another plate to seal this section. So essentially, you will put another plate on the top to restrict any entrance of external air. So then you just maintain anaerobic condition, and in the similar manner, you'll rotate the disc to do this operation. And essentially, in a reactor scheme, bigger scheme, we'll always have a settler after this reactor, because dislodgement of bacterial biofilm is very common. So whatever water you will get from this system, you will have some biofilm in the water. So just to remove this biofilm, you will have a settler immediately after this reactor system. Is it clear now?
Yes, sir, it is clear. Sir, what are the purposes for which these kinds of systems are used?
Because in a given volume, if you need to have a suspended cell system, in this within same volume, you cannot have that much concentration of biomass. So biofilm is essentially increasing the biomass density, and you can get higher biomass concentration in a given volume. So if you get higher biomass concentration, again, just go back to the earlier formula mu x. If you get higher biomass, your reaction rate will be faster. You will get higher conversion. So if you get higher biomass concentration and if you get faster conversion, obviously you will need less amount of time to treat certain volume of water. And for that reason, your reactor size will also get reduced. So in suspended cell system, for treating 1000 liter, you may require a reactor volume of 800 liter. Whereas for attached cell system, you may require 200 liter reactor volume, because reaction rate could be 5 times faster. So these are different, again, scales of similar kind of reactor. So here the bacteria, they sometimes we also use fungi. So a different color fungi could be there. Here you can see green color. Here you could see some black color organism. And when you take this, if you just scrap the surface and see under scanning electron microscope, you will see this kind of structure. So it is actually a biofilm obtained from a fungal bed reactor. So this film will actually show a mat kind of structure.
Sir, for seeing this, can you go to these kinds of structures? Can't we use a normal microscope, like a compound microscope, which we use normally in the lab? Or like, why do we need to use a scanning electron microscope for this?
So in compound microscope, with that resolution, you will not be able to distinguish, or it will not be able to have clear surface morphology of the film. So if you need to have clear surface morphology, then you go for ACM.
So, sir, it means like fungi, the filaments of fungi are of nano scale, nanometer scale.
No.
Something like.
No. It is nothing to do with only nanometer or micron size. here also, if you see the scale, it is given 20 micron. So it is not in a nano range. But when you talk about observing the surface in your compound microscope, it will not be that clearly visible compared to electron microscope. So when you observe anything under electron microscope, the picture clarity will be much higher. So you can actually get higher resolution picture, and you will get much clearer picture of the surface. So when you use compound microscope, you can see some sort of bacterial growth. I mean, sorry, the fungal media there. But if you really want to have very clear and high resolution picture, then you have to go for this scanning electron microscope.
Okay, sir.
Now, one of the is most widely used and common design is upflow anaerobic sludge blanket. So here you can see different granules are actually forming a bed, and all this granular sludge are actually self-immobilized granules. So each of these granules are made up of different kind of bacteria. So they will try to stay together and they will form a blanket kind of structure at the bottom of the reactor. Now, whenever you supply certain amount of wastewater, entire bed will try to move up. So it will move in upward direction, and then again it will fall down because of gravity, because of the mgh. So and as it will work like a moving bed or moving blanket kind of system, that's why it is called upflow, because flow is in upward direction, the system is anaerobic, and the sludge blanket is essentially um, helping degrading the wastewater. That's why this reactor is known as upflow anaerobic sludge blanket reactor. So in this reactor system, methane will produce. So methane will get trapped in the gas trapper, and then in the gas trap, methane will get separated, biogas will get separated, and the liquid. So the space between. So design is made in such a way that very small space will be available in between the gas trap and the reactor surface. So this granules, they will actually get trapped here. They will not be able to go out of the system. Only liquid will go, and gas will get separated in this cone, and all solid granules, they will actually get trapped here, and ultimately they will again, due to gravity, again come back to the main reactor environment, and you will get almost treated effluent from this outlet, and gas will be collected from the gas outlet. So this kind of reactor is very common and widely used for anaerobic treatment system, and the biogas generated from the system can also be used as a fuel. Only disadvantage is the process is quite slow. It is not as fast as your or efficient as your ASP, activated sludge. But advantage is you are not using any aeration, so operation maintenance cost is very low, and also you are recovering energy. So essentially, you can generate certain amount of revenue from the system, and the biomass you get there in the reactor, after a certain period of time, this biomass can be used, because these are all digested biomass. That biomass can be used as a fertilizer. So you have multiple benefit with this reactor system. Only disadvantage, system is very slow, and also startup time is significantly high, because you need to get substantial amount of biofilm thrown in the reactor first, then only you will to start the reactor. So as the startup time is very long, so failure of this kind of reactor is very, very problematic for the operator, because if you have any failed reactor system, reviving the reactor will take substantial amount of time. So this reactor is quite sensitive, very performance is also not as efficient as ASP, but advantage is the operation cost is very low, and also you generate revenue by producing biogas and also the fertilizer after digesting the granule. Okay, so I'll stop here today, and then in the next class we'll discuss about more on this USB and a little bit about aerobic fluid bed reactor. Then finally we'll just show other configuration of unit operations in the process.
Sir, in our department also like research is being done on these kinds of biogas producing systems and things like that.
Yes. Then we also develop in our lab also develop different kind of reactor configuration. So I'll show one of the design we have developed here, and maybe in the next class.
Yes, sir. That will be better.
Okay, sure. Okay, any other question? Okay then. But why this attendance is so low? Should I just keep little bit more weightage to the attendance then? In the class, I could see almost 67 or 68 students are there, but registered in this course. But if I get only 10, 12 students in the class, then I'll just record the lecture and put it on the web.
No, sir, recording lectures will not be better, but like we cannot discuss anything if we have some doubts.
I completely agree. For that reason only, I also prefer to have, because this course I am teaching in online mode for last 2 semesters. I could have actually kept all the old lectures, recorded lecture, and then ask you to see that. I'm deliberately not doing that because I prefer the interaction. But this is not the way to respond. Only 7, 8 students or 10 students are attending the classes. That is not fair. And if somebody can give justification that they are engaged with some other activities, then people can understand. But they are not doing anything. This period you're supposed to just study. So only option is to just give more weightage to the classes then. So we'll try to have.
We have already like 40 marks for class participation, etc. So maybe that in that way, that maybe you can do like 5 marks for attendance or something like that.
Deliberately, I was not doing that because many students will have issue with internet connectivity or maybe COVID problem also. But if anybody has such kind of issues, they should inform us, and then we'll try to resolve that issue also. So whenever you have any problem, we also have solution to that. But this is not a good practice that they will not attend the classes. That is not actually acceptable. So maybe in the next class onwards, immediately after the end of the lecture, I'll just conduct a quiz, small quiz. So every day we'll have quiz. Let's see.
Sir, like what is the current evaluation policy? Like in the first class, you mentioned that 30 marks for minor and 30 marks for major and 40 marks for class participation and assignments.
Agree.
Right.
That was considering good participation from the student in the class. But if students are not coming to the class, then we have to change the policy.
Yes, sir. So isn't it that already the class participation is mentioned for the evaluation policy for the evaluation of the course?
Now, see, class participation means, as I told you, I'm not going to keep any marks for attendance, only attendance. Suppose I am conducting a quiz in the class, definitely that will come under that category, that within 40% quiz marks can come, and we can give you assignment. Obviously, then you can get some marks in that also. But if we feel that students are not actually responding, then instead of keeping 30-30 for minor major, I'll keep a little less in minor and major, and I'll keep maximum portion for the class participation then. Instead of 40, I'll raise it to 60 then. My logic is those who are attending the class, they should get maximum advantage. Who is the class representative here, or maybe he is not here today? So one of you can actually, yes, tell me.
Perhaps he is not here, I guess.
So one of you can take a stand, maybe send the mail to the group that those who are not coming to the, means it will be the, maybe in the next level, next day or next lecture class, we'll just try to have a quiz test. So automatically that message will go to the entire class then. And specifically 2020 entry students, if you are not attending lectures every day, I'll actually be a little bit harsher to your evaluation, and definitely I will not check your answer script leniently, and you can expect a little bit lower grade compared to 19 students. Because if you have that saying that you have free time this time, that's why you have taken this course, so then I expect seriousness from your end also. So all 2020 entry students, whether it is from computer science or from DBEV, your attendance should be 100%. If you have any issue, just mention it to me. We'll consider that, but you should attend all the classes. Okay, let's see you next week then.
Thank you, sir.
Thank you.
Thank you, sir.